Hello. Hello, welcome everybody. Welcome to the stream. Hello to you all. Hello, A and Kevin and Clockwork and Arch and the Clockhead and Clockwork. Surprise Pants. Hello, welcome. Today we're going to be talking about everyone's favorite morbid fascination, prions. But before we do that, I wanted to give two special shout outs. One, again, to Emily. Happy birthday, Emily, if you are going to be watching this stream. And also, I wanted to shout out. I wanted to shout out Draco, who is the first NK Cell member. So I was maybe a little over ambitious with what I was promising members in terms of my initial membership stream. And so I recently gotten rid of some of the tiers and really pared back what I was prepared to do, because I found that like fulfilling some of these member role incentives was maybe a little too much for my content schedule. But I left the NK Cell one up there and just changed the description to I will acknowledge you as an absolute Chad, just in case someone wanted to throw that much money at me. And so here I am acknowledging the absolute Chad that is Draco. Thank you very much for the support. Very much for the support. Welcome DC Iron Wolf. Welcome to the stream, talking about prions today. And while people are filing in, nice shout out. True. As people are filing in, I just wanted to tell a quick anecdote before we get started about realizing my age and place in this universe, or at least in the scientific ecosystem. I was taking instruction from a postdoc on how to do this technique that I've never done before, and I had always viewed postdocs as being sort of the adults in the room. Of course, ultimately, the biggest adult in the room is the PI, the head researcher, the leader of the lab. But if you've been in a lab, you know that they are usually not the first person that you would go to when it comes to help with troubleshooting, sort of on the ground, in the lab kind of stuff. PIs are great for providing an overall strategic vision, but in terms of, and they'll admit this themselves, a lot of them will at least, they haven't been in the lab for a very long time, and so postdocs become sort of the most senior authority on how to actually do things. And this postdoc was showing me how to do something, and I had realized that I am the age of a postdoc at this stage of my career, and that, you know, this vision that I've had of postdocs as being the adults in the room, I'm like, oh no, I'm adult age. I am adult age. And that was a really devastating realization, to know that like these people that I'd looked up to scientifically for the majority of my career, these experienced, talented researchers, I am now not the same level because I don't do active research, but I am now at the age of those people, and so that's a bit more devastating than I had expected. Although the postdoc did ask if I was doing my master's degree, so I can still pull off maybe looking a little less haggish than I actually am, which is why I wear sunscreen. I told this anecdote on a previous stream, but I had been mistaken for an undergraduate, and I'm not humble bragging, I'm flat out bragging, and that's what motivated me to, you know, overcome the discomfort of applying cream to my face, and now I am an advocate for wearing sunscreen, because there's only so many more days that will happen, and someday in the future that will be the last day I will ever be that I will ever be mistakenly recognized as an undergraduate, but I want to push that off as far into the future as physically possible. Absolutely terrifying. Yeah, I know. The prospect of actually being the adult in the room is terrifying. Okay, so let's talk about prions. Okay, here we go. So today we are going to be talking about this paper from my beloved PLOS biology journal. Is open access, open source, not open source, open access journal. So I can actually show their data from the wonderful, amazing Lindquist lab. So this paper is called Prion Switching in Response to Environmental Stress. So this title, I think, actually undersells how crazy this story is about to get. And there's going to be a lot of background when it comes to appreciating how insane this paper is, or at least the research that surrounds this paper is. So bear with me, there will be quite a few slides before we get into the actual meat of the paper, but trust me, this is a story you won't want to miss out on. Okay, so hopefully if you are in the stream, you have watched my prion video, but just in case you haven't, or if you need a reminder, a prion is any protein that when misfolded can force other prions or other proteins to misfold. So here I have a very simple illustration of a properly folded prion, major prion protein, and a misfolded prion protein. And when these two come into contact, that properly folded one will misfold and aggregate, and that leads to all sorts of unfortunate things like neurodegenerative diseases. So yeast make prions as well, but just so that we sort of anchor this into something that we as humans are more familiar with, I'm going to talk a little bit about the major mammalian prion protein. So this is the prion protein that many of us are familiar with because of how scary that the potential for this protein to be is, right? So when this particular prion is misfolded, you get things like CJD, Kuru, fatal familial insomnia, mad cow disease, etc., etc. This is in its healthy form, and people took issue that I mentioned that there are healthy prions. This protein is called the mammalian prion protein. So when this protein is properly folded and not exhibiting the properties of a prion, it's actually doing something for our neurons, but when it is misfolded, you get disease. So something that's interesting to note, and something that I want to re-emphasize from that video, is that in terms of conservation, there are dozens of mammalian species that have confirmed to have this major mammalian prion protein, as well as some birds. So a major question that we like to think about is why this prion has stuck around. Now, it's true, and as many comments in my prion video have pointed out, that nature doesn't necessarily cull proteins that are not useful, so as long as they don't have a disadvantage. Well, it's also true that when things are generally well conserved, that is generally true for a reason, even if it's not very obvious. So chat is stretched and weird on stream. Yeah, I had a little bit of trouble with managing chat on OBS. I think OBS had updated and it's been kind of weird ever since. Let's see if I can change that. Oh yeah, it is cut off. Hold on 1 second. I might not be able to fix that. Oh, that's so unfortunate. Yeah, I was messing around with the chat box and it doesn't appear like it wants to cooperate today, which kind of sucks. So I'm sorry, you're just going to have to deal with that until I can get it fixed next stream. Yeah, I know, I wasn't expecting the OBS update to like break this in this particular way, so I apologize for the chat being cut off. I can't even really move it, so maybe I'll just hide it if that's okay. I mean, because there's like a playback, and it might be a little distracting if the chat is cut off. Okay, so that's sort of the context for why we care about prions, at least in the mammalian context, right? But the major mammalian prion protein is something that is mostly made in neurons. And what's interesting is microbes like yeast that don't have brains or neurons also make prions, not the mammalian prion protein, but proteins that exhibit prion properties, I should say. Okay, so the question that's interesting here is, what exactly are these yeast prions actually doing? Okay, so yeast actually make a ton of prion products. So you should see, if you've seen my prion videos, some familiar faces, including this Het S prion that has a positive role in making sure that you don't get careless mixing of different yeast strains. So this one's important for something called heterocheiron formation between incompatible strains. Basically, if two yeast of different strains, incompatible strains, mate, this is basically a self-destruct button to ensure that you don't get this kind of careless mingling. The protein that we are going to be talking about today, however, is this protein over here. The healthy protein form is called SUP35, and the prion form is called in brackets, PSI+. So that's just sort of the nomenclature to help us understand the language that we are about to encounter here. Okay. So prions are very weird, and I remember learning about this when I was writing a grant to study prions back when I was an undergraduate. And I unfortunately never got that grant. It's very hard to get grants these days, or at any point of time, really. But I wanted to couch this upcoming discussion in sort of what I find to be fascinating about prions, that they seem to go against everything that we take for granted in molecular biology. And one of those is the fact that they are potentially, depending on who you ask and how you define things, again, language is only so powerful, but they are potentially an exception to something called the central dogma. So if you are a biologist, you know that there is this thing called the central dogma that describes the one-way flow of information from DNA, which is this molecule over here, to RNA, which is this intermediate over here, and then eventually to proteins. So the central dogma states that this is sort of the direction by which information flows. So prions actually represent something that can sort of not interrupt, but runs not according to this kind of plan. Because, as you see here with this now prion accurate, oh wait, hey, that's weird. Whoa. Okay. 1 second. Wouldn't be a presentation without a tiny bit of scuff. Fun little thing that happened. So we'll present this. And we're back to the beginning. Okay. You know what? I'm just going to continue blushing. The presentation is misfolded. That is very true. Okay. All right. So prions are a potential weird exception to the central dogma, as we'll see here. So prions, when they are misfolded, cause healthy prions to misfold. And so the information that is carried in their own reproduction, the reproduction of prions, is not encoded in the DNA, or it doesn't have to be encoded in the DNA. Prions can reproduce simply by coming into contact with correctly folded prions. And so there's this weird adjacent counter flow of information that can go from protein to protein instead of DNA to protein. So that is a very interesting exception to the rule, and of course I am fascinated by exceptions to the rule. Yeast prions are especially weird. So here is sort of the pitch for this paper. This idea that I always kind of assumed existed but never really thought about. So this is a segment from their paper that says evolution depends on the manner in which genetic variation is translated into new phenotypes being traits or traits that you can observe. There has been much debate about whether organisms might have specific mechanisms for evolvability, which would generate heritable phenotypic variation with adaptive value and could act to enhance the rate of evolution. So this idea of evolvability is not really new. There are viruses, RNA viruses especially, that have a high base rate of mutation, partly due to the fact that some RNA viruses carry polymerases that are not very good at copying DNA very accurately. So you get this baseline mutation rate that is higher, that allows RNA viruses to mutate at a higher rate to adapt quicker, you could assume. So there's a question in chat. Why do misfolded prions misfold healthy prions, but not vice versa? So when prions misfold, they adapt this very nasty sort of shape, these beta sheets. So we go from a very alpha helical structure, as represented up here, to a structure that is beta sheet heavy, and those beta sheets stack. And then every stack of beta sheets, you get another layer of intramolecular forces. It's basically just very difficult. You can kind of imagine this alpha helical floppy prion as being like putty, and then when it collapses and misfolds, it becomes like solid, is one way you can think about that. Okay. So this idea of evolvability is, again, not unique in the sense that we know that RNA viruses have a higher rate of mutation and can adapt quicker to, or these, you can draw a line to them adapting quicker. I should say it's correlated. But the thing that we're going to be talking about is whether or not yeast can trigger evolvability. And that's interesting in a mechanism that has nothing to do with baseline genetic mutation. So I want to illustrate this idea of evolvability a little better, right? So you're yeast, right here. You're a population of yeast, and there's this environmental stress. Maybe there's no food. Maybe there's a ton of salt that appears out of nowhere. Maybe there's an antibiotic that's been introduced to your media. And you have two options. Either you die or you survive, right? And so, no, let me move myself out of the way over here. And so, next slide. If you have a significant amount of environmental stress, maybe your media is being cooked at a very high temperature, you're going to get a significant proportion of your initial yeast population going to death. And so there is a chance that nowhere in this population of yeast exists a yeast with a particular mutation that will save you from being cooked alive. So the idea of evolvability is that, well, if none of us are going to survive anyway, maybe we should have a mechanism that just scrambles our DNA and increases our genetic variation. Yeah, and so Clockwork said, when stressed, panic and hit the Darwin button. That's a great way to imagine this idea of evolvability. But the weird thing is that that's not even, okay, you know what? I'm not gonna, I'm not gonna bias you. I want this to be exciting. But that's essentially what the idea of evolvability is. When there are tough times, to push the Darwin button and evolve as much as you can, or mutate as much as you can, so that you can evolve to overcome some significant stress. So evolvability may enable more yeast to survive really extreme tough conditions, like a break glass in case of emergency type situation. So to illustrate sort of mathematically what this might look like, or with images, is let's say you have a population of yeast, and this population of yeast gives rise to all of these daughter cells at some point. If there's no evolvability and you get a deadly challenge, then okay, all of these yeast that are identical to the parent yeast, they all die. That makes sense. None of these yeast have mutations that would help them tackle this deadly challenge. But if you have a situation with high evolvability, and I couldn't think of a different way to illustrate evolvability than to crop PNGs of wigs and stuff and put them on these yeast. But if you have a mechanism of evolvability that might allow some of these yeast to develop mutations that would allow them to survive whatever deadly challenge there is here, right? Okay, so here's the hypothesis of this paper, right? And before we get into what they actually do, let's just, in order to appreciate how amazing this paper is, let's come up with a hypothesis here, or a method of testing. So the hypothesis is that the yeast prion, PSI, increases evolvability. How would you test this? Slay yeast slay? Yeah, it's true. Very girl boss yeast on there. Yeah. So how would you test whether or not the yeast prion increases evolvability? So A says remove it. So we're going to need a plan that's a little more complicated than remove it, because removing it doesn't necessarily test evolvability depending on what you measure. So removing it might be a good start, but what would you measure? Separate yeast populations into control and experimental groups. That's always a good start to any scientific method, but how would you test evolvability? How do you remove it, says Ramsey? You could knock it out. There are genetic engineering methods that you could do, so I'm not concerned about that. Gradually increase stress on experimental group and sequence the genome of each generation and measure variance. That is certainly an idea. Use some kind of reporter plus some kind of directed evolution campaign, GFP expression plus facts. So D7 clearly has some molecular biology background. So a reporter is a gene that will allow you to determine whether or not a protein is being made, and some kind of directed evolution campaign is basically putting a restraint on the yeast. So maybe growing them without glucose as a source of sugar, for example. And then fact sorting, D7 is recommended, is a method by which we can separate cells into cells that do make prions and cells that don't. All very interesting ideas. I think the idea that I had is most similar to the one that Clockwork had come up with. No problem. I love the opportunity to sort of talk about these more advanced techniques. My first hunch is to create an environment that has regions of increased harmfulness, like that one famous E. coli evolution video. I don't really know which one that is, but we're all coalescing around exposing these yeasts to harm and then measuring some output of evolution afterwards. So this is what I've come up with. So much like Clockwork had said, I have two groups, and so the nomenclature here is that capital PSI plus is prion containing, and then PSI minus is no prion, and subject them to a gauntlet of terror. So, a lot of things that would kill yeast, and then I would do two things. I would check viability. So the hypothesis is that evolvability should be improved, and so the prion containing yeast should survive better because they are evolving to overcome challenge. And then two, sequence yeast DNA, much how several chatters have already suggested. So prion containing should have interesting mutations, right? So we're coalescing sort of around the same idea, even though we might come at it from slightly different approaches. However, the yeast prion PSI is too weird for this approach to work. It's too weird. And that was sort of a devastating thing to learn as I was reading through the paper, because I had this notion that I was like, okay, I can predict where this paper is going. We're going to be measuring evolvability. We're going to be sequencing DNA. That's not what we're going to be doing. And I think that is really, really, really surprising, because how do you have evolvability without mutation? Huh? That's weird. How do you have evolvability without mutation? We're not going to be sequencing DNA here, but we are still going to be measuring evolvability. And I want to just sit in that confusion just a little bit, because we are measuring evolution, but it has nothing to do with DNA. Okay? Weird, I know. And it has to do with what I mentioned at the beginning of the stream, that prions are violators of the central dogma. Okay, stay with me, okay? It's going to make sense by the end of the stream. And this is why I'm so excited to talk about this, because the idea that you can have evolution, or the appearance of evolution, without changes in DNA is crazy, just bonkers. But we're going to see how this is possible. Charles Darwin is rolling in his grave. That's true. Actually, it'd probably be Mendel. Darwin had no idea what DNA was, basically. Changes at the transcriptome level, no. Well, yeah, yes. Okay, yes. Okay, Clockwork's getting a little ahead. Changes in transcription. We'll see. So it has something to do with changing proteins post-production. No, clockwork is correct, and we're going to see how that happens. Good guess, though. Okay, so what does PSI actually do? So I've underlined the point that is interesting. This is the same segment as before, so let's just start off where we have new information. Capacitor systems, which allow for the accumulation of cryptic genetic variation and release it under stressful conditions, might provide such a mechanism. Capacitor systems. So something that is a capacitor, if we're talking about electrical engineering, is something that can hold charge. So PSI, they are arguing, represents a sort of genetic capacitor that can hold variations in genetics and release it under stressful circumstances. So what the heck does that even mean? Let's continue to read. Oh, sorry. Is my cursor? Yeah, there it is. In yeast, the Prion PSI exposes a large array of previously hidden genetic variation, and the phenotypes it thereby produces are advantageous roughly 25% of the time. The notion that PSI is a mechanism for evolvability would be strengthened if the frequency of its appearance increased with stress. That it is a system that mediates even the haphazard appearance of new phenotypes, which have a reasonable chance of adapted value, would be beneficial if it were deployed at times when the organism is not well adapted to its environment. That's a lot of jargon, but I want to keep this idea of a capacitor system in mind. And if it's not useful for you because you don't know or are not comfortable with electrical engineering, just think of PSI being the break glass in case of emergency, where once it's deployed, you break the glass on some hidden genetic variation that you didn't know existed, and that helps them overcome stressful challenges. And I know that seems crazy, especially if you've come from a biology background, but we're about to get into it as I get into the theoretical background of this model. Exciting hypothesis, exactly. So PSI is what's called a nonsense suppressor, and that's not just what I wish from Santa every year. Bada bing, bada boom, hilarious joke, makes it so that I don't have to respond to certain emails. That would be nice. But what is a nonsense suppressor? So in order to understand that, we first have to take a look at the healthy, correctly folded version of the PSI Prion, which is sub 35, whose picture I am showing on the screen right now, right? So this is sub 35. So what does sub 35 do? And of course, just as a note, we can appreciate these long spaghetti strands of unstructured protein. So for those of you who have never looked at a protein structure before, you have these ribbons and these beta sheets, these arrows, and these represent sort of more stable configurations or more stable areas of the protein. And then you have these long, thin lines that represent this very flexible spaghetti that is characteristic of an intrinsically disordered protein, a protein that is very floppy, and prions start off in this floppy way and sort of collapse into a misfolded form. And then these floppy bits turn into these arrow bits, making them a lot harder to fold. So what is PSI doing in the healthy form? So its normal healthy form is called sub 35, and this is called the eukaryotic translation release factor. So what this does is it helps the protein production process stop when it's supposed to stop, right? So just to get all of our nomenclature on the same page, healthy version is sub 35, the prion form is PSI plus, and the non-prion is PSI minus for the future, right? So sub 35 is a factor that binds the ribosome, and it basically recognizes when there's a stop codon, and when it hits that stop codon, it helps to disassemble the ribosome and stop the process of elongation. So if we go from the central dogma, DNA becomes mRNA, becomes protein. But in order for a protein to be made correctly, there is a code within the mRNA that tells the ribosome to stop making that protein. So down here we have a ribosome, or a cartoon of a ribosome. And you can see here that as we continue on in these steps, that these tRNA bits are being gradually added. And so you get this elongating chain. So these balls up here that look like Dippin' Dots, these represents the amino acids that are stitched together within this complex called the ribosome, right? And this line down here represents the mRNA that contains the code to make that protein. And so here I have a string of DNA, This is just one that I made up, and this gets transcribed into mRNA. So all that is a change between T and U, and so I don't remember if I have to do this for the complement, but you know, if I get this part wrong, that is because I don't have this process memorized at the start. But let's just say that it's a one-to-one and I don't have to worry about reverse complement or anything like that. I just realized that that could have been a factor, but I'm too tired to remember if that's true. And so the mRNA sequence, having those T's be removed, is going to have a stop codon. And so UAG is one of these stop codons. So if we look at the sequence of amino acids that are stitched together to make this protein, we have a methionine, which is the start codon, followed by, I'm gonna have to do this from memory, a methionine stitched with a glycine, proline, phenylalanine, lysine, glutamate, tyrosine, aspartate, glutamate, and then followed by the stop. And so SUP35 recognizes UAG and terminates protein production. So you cut off the protein at the right time. So we see here there are still DNA sequences after the stop, GGG, GAA, and CGA, but these DNA bases don't get made into protein eventually, right? Correct on all counts, thank you very much. Okay, so yeah, so SUP35 tells the ribosome it's time to stop when it recognizes the stop codon. So nonsense mutations, the nonsense part of nonsense suppression, nonsense mutations introduce stop codons where they're not supposed to be, turning the protein into nonsense. So you have this long protein chain, it has this specific sequence and this specific shape, and if you have an early stop codon, that can turn the entire protein non-functional, which would be very unfortunate. So here, we have TAT, which is translated to UAU, which becomes tyrosine. But if you have, say, some UV irradiation or something that causes a single base pair mutation, you can turn from TAT to TAG, and then that becomes a stop codon, right? So if you have an early stop codon, then this protein is likely not going to be functional anymore. So that's really unfortunate. So a nonsense suppressor is something that ignores stop codons. Okay. So PSI is a nonsense suppressor. So SUP35, non-prion healthy protein, stops protein synthesis when it detects a stop codon. And in the prion form, it actually removes SUP35 by aggregating, right? We know that misfolded prions cause healthy prions to misfold and then they start aggregating. So the prion form of this protein pulls SUP35 from, the cytoplasm and makes it unusable. So you no longer get this protein that helps the ribosome know when to stop. And in that case, you get proteins that are ignoring their stop codons, which is kind of crazy. So I'm going to stop there. I'm going to take questions from the chat because this is really important background to know so that we can further understand the story in the future. Okay. And I'm going to take a sip of water. So if we understand what a nonsense suppressor is, then we are going to be good to go. So are there any questions from chat? Okay. Alrighty. Okay, so I've had my sip of water and we are just about ready to continue. Am I going in the right direction if I ask what pressures causes stop codons to get inserted in an organism's genome? I don't really know too much about the selective pressure on stop codons, so I would not be able to answer that. I feel like an amateur microbiologist listening to this. Technically, we might be more in the realm of just molecular biology because we're kind of in between microbiology, dealing with yeast, but also talking a lot about biochemistry with proteins. If the protein synthesis isn't stopped, wouldn't the protein get longer and longer, eventually until it hits another stop codon? So just because SUP35 isn't there doesn't mean that it just kind of helps the ribosome recognize that there's a stop codon. So it's not 100% of the time. You get leakage, basically. But that's a good question. That's why I always say that biology is a spectrum. It's never all or none. It's always something along the spectrum, right? Okay. All right. So let's continue. So basically, the PSI prion ignores stop codons and continues to add absolute genetic garbage on top of the protein, which is, as you can imagine, not very helpful. Wondering if okay, no. Okay. All right, let's continue. So what do we mean by PSI can act as a genetic capacitor? PSI actually helps to ignore stop codons, which unlocks cryptic genetic diversity. Cryptic genetic diversity. Okay? So here's the thing. You have DNA, and a lot of that DNA is non-coding, but a lot of that DNA is also coding. So what do I mean by that? So a lot of your DNA does code for things like protein, but then in between those protein coding sequences, you have a lot of DNA that for many years people thought did nothing. Turns out that's not exactly true, but you have, broadly speaking, coding DNA and non-coding DNA. And in evolution, you have a chance that certain sequences get mutated, or rather, if we just take a look at your chromosome, if you get hit by UV light in a weird way, then that can cause mutations. You can get weird things happening during replication of DNA that can introduce mutations. The point is that your DNA is always mutating. It's up to the immune system to make sure that those mutations don't turn into cancer, but the basic idea is that from you to your child, from your parent to you, there is some level of mutation that occurs. And across evolutionary time, there's been a significant investment in making sure that you survive. And so a lot of the protein coding regions that you have are what's called highly conserved, or relatively highly conserved. And what that means is my hemoglobin should look like your hemoglobin. If they look that different from one another, then one of us is going to be in trouble, right? We need to make sure that there is selective pressure on keeping the proteins that we need to work functional. But the coding sequences between the protein coding regions, the ones that are more regulatory or for spacing reasons are there, spacers in between protein sequences. It's not that they're not important, but it's that their exact sequences don't matter as much as the part of the DNA that codes for protein. And so in these regions, after the stop codon, you get a lot of low conservation. So you get a lot of variety. So in here, I have outlined in red. GGG in yeast 1 can become GTT in yeast 2, and then GTG in yeast 2 was CGA in yeast 1, right? I see. It sounds like open reading frame is lengthened to the next stop codon or until the ribosome falls off the mRNA. Exactly right, Resta. That's exactly correct. Does this factor in with anything Alzheimer's research related? I'm not ready to talk about Alzheimer's. I'm going to stay with prions because I don't want to get any of that information wrong. But people do talk a lot about prion-like diseases and Alzheimer's in the same breath, but maybe one day in the future. Okay, with all of the information here, let's walk into my mind and react or try to understand why I found this hard to believe. So here is the claim. The yeast prion PSI can improve evolvability by ignoring stop codons to generate more protein diversity. Daughter cells inherit the PSI and continue ignoring stop codons. So that's how they violate the central dogma. They inherit these mutations, but it's not because their DNA is different. It's because they inherited the PSI. When the yeast split, right, they bud off of each other. So if the daughter cells get the PSI, then they also inherit the ability to ignore stop codons. Why do I find this hard to believe? Why am I skeptical that this prion that ignores stop codons is actually a good thing? Why is this so confusing to me? Not exactly an easy, maybe it doesn't confuse you at all, but it definitely confuses me. So why is this confusing, chat? Does this, especially the biologists in chat, you might not agree with me feeling skeptical here. But why might I have felt skeptical? Does that make sense? Give chat a second while I drink a little bit of water. At least chat on my end has stopped. I don't know if y'all are chewing on it, or maybe chat isn't updating. Let me go ahead and check real quick. Okay, no, chat's fine. Okay, A very bravely saying that they don't know. That's fair. How can individuals survive that their translation machines are pumping out these big weird peptide clumps? Yeah, thinking hard. Yeah, it's a tough question. I'd imagine that the new proteins produced as a result of these prions would not necessarily improve the daughter cell's fitness. That's getting closer. As usual, Clockwork, I think, is on the same page as me. Guy G seems to get it too. The mechanism should create very long proteins, not functional proteins. Right. Proteins are evolved to do their thing very particularly. Evolution has honed them to be very good at their jobs, and usually when you add a whole bunch of random amino acids at the end of a protein sequence, it doesn't function quite as well. So generally, ignoring stop codons is a very, very bad idea. Very, very bad idea. Ignoring stop codons, not a good idea. So that's why I found it hard to accept, but I'm going to accept it anyway for the sake of the rest of this paper, right? Because here's something that I want to model for people who want to be scientifically literate, is walking the line between skepticism and cynicism, right? I am, on one hand, skeptical that ignoring stop codons could ever result in a more functional organism, but I am also acknowledging that I didn't read the paper that showed that they were. I'm also not ignoring that, my scientific instinct. I'm merely stating that this is a claim that is hard to believe, but I'm going to assume that it is true because I'm reading a paper and I'm being like nice. You know, you always want to give the authors a chance to win you over. You never want to come in and just be grumbly and cynical all the time. But I am stating outright, I'm being true to my feelings. It feels wrong that ignoring stop codons could ever be a good idea, but okay, I can see how this could work, that maybe you need that extra bit at the end of a protein, and across millions of yeast, maybe one of them will survive some terrible condition by this very desperate Darwin button, as Clockwork had said. Is there a mechanism that happens after translation that can help these peptide sequences become useful? I mean, maybe they'll luck out and be post-translationally modified in a way that works out. I don't know that there's necessarily a guided way that becomes true. The daughter cells could inherit only a small fraction of the prions, so they survive long enough to pass them on. Yeah, and that's the interesting thing, is that this is potentially only a stopgap, because once those daughter cells lose that prion, they also lose the ability to ignore stop codons. So it's an interesting temporary break glass in case of emergency type situation. Are they random in this case? So these sequences that are after a protein coding region that are not coding for protein, they're not random per se, but they have a lot more variation. So we're kind of playing gotcha here, right? We're just doing the odds that something in this more variable region of the DNA could make something, if we allow it to, that could be useful. These new random peptides need not themselves be adapted to begin with, though, just serve as substrates for selection when these yeast come across hard times, according to the story that the authors tell. I mean, yeah, but I mean, to me, that's not really different. Like, immediately adaptive and eventually adaptive, to me, feel the same. It's just kind of hard to believe. There are a few breaking glass emergencies in genetics, non-homologous end joining, for example. I think that's what that stands for, but I'm not entirely sure whether or not that is the case. Oh, Jim the Evo. I think I recognize you. Oh, you wrote for Kierkegaard, right? or SciShow or somebody. Awesome. Welcome to the stream. Very happy to see you. That's awesome. That's awesome. Very exciting. Hey, say hello to Jim, everybody. Great content. Also low-key jealous that you got to write for them. Very cool. Very cool. Okay. Okay. So here's the hypothesis here, right? the yeast prion PSI increases evolvability by ignoring stop codons. How would you test this? Now that we have the full picture, how would you test this experimentally? And while you are thinking about that, I will address Simon's question. Wouldn't the random peptide sequence not even likely code for functional proteins, as the reading frame wouldn't be preserved in the non-coding region? Exactly. That's why it's like I think the authors are quite careful in the sense that like this is not, I think, a great idea for the yeast to do, right? This is purely out of desperation. Like if you knew that you were walking into a pit of lava and there was like 0% chance that your human body right now could tolerate that environment, this is a extremely desperate tactic, we could call it, to shake things up to the point where you could get something that could be something functional. I don't want to like dismiss D7's point about serving as substrates for selection, and I also didn't really come across that in the paper. I maybe skipped over that as I was reading through the slides. So I will acknowledge that if I give the paper another read, maybe I will have caught that. But I don't recall seeing that as an argument, although I could be wrong. So just want to cover my ass there. Okay, so how would you test this? So the yeast prion, they're arguing, increases evolvability by ignoring stop codons. So to me, that is much harder to think of a strategy, but it's cool. It's cool to think about how you would do that and compare it to what the authors ended up doing to sort of gauge the contribution of this particular paper. No idea. Maybe adding random stop codons in the original genome and see what if you can regain function. Interesting. I think that that might be a quite laborious method. Into the original genome and see what if you can regain function. So the idea here is that it's good that the prion causes you to ignore stop codons, because that means you can get wildly different proteins out of your genome. If stop codons are ignored, the resultant protein should have higher molecular weight. That is true. The hypothesis is that it's increasing evolvability, though, so that's the part that we want to test, and the mechanism is by ignoring stop codons. So we already kind of know that it ignores stop codons, but is it increasing evolvability because it ignores stop codons? That's kind of the interesting question here. I'm going to take one more sip of my water, and I will march on. I am now like a little nervous that there are quite a number of smart biology people, and please don't feel bad if you need to correct me. I want to make sure that I nail it in this presentation, just so that, because there's a record of this online. Alrighty. Okay. I've been around since the beginning. It was easy to get involved with this stuff when I started. Yeah. Jim, we should chat. Maybe I shouldn't be trying to network live on stream, but I would be interested in chatting one day. Okay, so here's my proposed experiment. To check whether prion-mediated nonsense suppression improves evolvability. So you get the two yeast, one that contains prions and ones that don't, and there are methods by which you can remove prions or grow yeast in a condition that discourages the formation of prions, subject them both to a gauntlet of terror, and there are a couple of things that I want to check, right? One, check viability. So in this gauntlet of terror, there should be yeast that make this prion that survive better. Of course, evolvability is not just a break glass in cases of emergency and then just die. That would not be very good. I would want to sequence all proteins with a handy-dandy mass spectrometer, so not sequencing at the level of DNA, because we know that the DNA in this case doesn't really change. Like, that's not what's changing here. What's changing is the mRNA, you could say, which is why Clockwork was correct about the transcriptome. So a mass spectrometer, as Resta is hyped for, is a very cool machine that breaks all the goddamn time, as I have learned as a graduate student. And it basically allows you to sample the entire protein environment of a given sample. So say you want to know, okay, what are all of the proteins that are in the blood? You would then prepare a sample for the mass spectrometer, send it off to the spec, and then you would get back a list of all of the proteins in that blood, or at least all of the proteins that the mass spectrometer is able to detect. It's very complicated. One day I'll probably talk about mass spec because it is a big deal, but just know it is a very powerful technology that can tell you the identity of proteins. But actually, that doesn't actually work in this case. It was my initial idea, but after thinking about it, mass spectrometry, I think, and mass spec people in the chat might be able to correct me on this, but you have to know what you're looking for beforehand. And since these yeast are spitting out random additions to proteins that, you know, the universe hasn't seen yet, mass spec might not actually work in this case. So it was my attempt at tackling this problem that might not actually pan out very well. Then I would sequence the yeast DNA to find out which codons were suppressed and where. Again, contingent on 2, that doesn't really make a lot of sense. And then 4, if I could identify which weirdo proteins corresponded to which stresses, I would then try to add those weird proteins back into non-prion containing yeast to try to figure out whether or not that's the thing that's helping them survive the gauntlet. And again, this doesn't work because I don't think you can do that with mass spec because of how weird this system is. But that's my best attempt. And all the more respect to the authors and this lab for tackling this problem in a way that makes sense. If you could do long read RNA sequencing, mass spec might still work. You'd see the random bits at the end of the express transcripts. Mass spec would then tell you if they're present at the protein level. That's true. So what D7 is saying is that if you could predict what the proteins would be, then you could essentially have your own little database of what proteins to expect. I think that would be cool. Oh, and Resta is in the chat, so maybe Resta can contribute here. So there are RNA sequencing methods that can sample the transcriptome of a sample, right? Right, Resta? Possible nowadays with PacBio IsoSeq. Very cool 3rd generation stuff. Interesting. Wasn't around 2008. Yeah, this is a bit of an older paper. That's true. So I know that you can sequence a complex population of microbes, so I would assume that this is similarly possible. I also don't know if mass spec was that advanced in 2008 either, so maybe this is my benefit of looking back with hindsight, but that's a very good point, D7. Okay. So I'm going to ignore that just for time's sake and continue. Beeping? Yeah, I think the garbage collector is outside. Okay, with all of that background, we are finally ready to talk about the paper. But before that, Ronan looks like he's an interesting question. They have an interesting question. Why can't you do an RNA sequencing experiment and compare wild type and experiment? So I would want to know the protein. So just because a transcript is made doesn't necessarily mean that the protein is successfully made, or whether or not they're, it's just getting information at the protein level is a lot more direct according to their hypothesis, but I think that's a legitimate line of inquiry that you could do. But in terms of the read-through, yeah, you would get that through mRNA. Yeah, that's true. You have a point, Ronan. As someone who likes proteins, I just typically tend towards wanting things at the protein level. That's my particular picky bias, but I think you might be onto something with maybe just doing the RNA level. Certainly a valid point. Okay, we're finally ready to talk about the dang paper. Again, from the Lindquist lab, the excellent Lindquist lab. A lot of prion in yeast stuff was done by them, which is very exciting. Okay, so here is a model for evolvability. So you have your yeast, and then you subject them to some environmental stress, and then you get this transition between this nonsense suppressor, this eukaryotic translation termination thing. You get that conversion to the prion, removing it from the cytoplasm, and that then suppresses the nonsense, and then you get survival. So I'm not saying that I agree with this model. I haven't read enough. But this is sort of the idea that we're trying to test. And so when you are thinking scientifically, it helps to draw something out like this so that you can clarify to yourself what parts need rigorous testing. And so the experimental approach here comes in two main parts in this paper. So one is to determine if the stress response genes affect PSI formation. So if we are making the argument that the prion protein PSI is something that reacts to stress in the environment, we should reasonably expect that genes involved in the stress response control the formation of PSI to some degree. That's a reasonable thing to expect. And number two, determine if stress causes PSI formation. So this is maybe something that is a little more straightforward. So if it really is regulated by stress, we should see stress cause PSI formation, as well as hopefully increase survival in some of the yeast. So this is a good checkpoint, because we are then going to dive into the data. But before we get into that, we should all make sure that we understand the science at this particular level. So I'm going to start a poll, and if I need to do a recap, I totally will. I'm just going to write understand while I take a sip of my water. MALDI-TOF is the same as protein mass spec. That's certainly an acronym that I have heard before, but I can't remember at the top of my head. Yeah. And something to keep in mind while we're pondering or looking at this experimental approach is, like, do you like this? Does this tickle your fancy? It's important to develop a sense of scientific taste too. I think it makes reading about science a lot more fun. Okay. It looks like we're mostly on board. But for those of you who said no, I'll just do it really quickly. So sup35 is something that tells the ribosome to stop elongating a polypeptide sequence, so stop making protein when it sees the instructions to stop making protein. So yeast need it so that they don't make infinitely long proteins that don't do anything because they're just garbage. And so when this protein, this sup35 protein, aggregates, it's no longer able to help with it's no longer able to help with recognizing stop codons. So you get something called nonsense suppression. And so the idea of this paper is that they want to test whether or not this prion actually improves evolvability by ignoring stop codons to unlock some hidden genetic variation. I may not have understood all the details, but I get the general gist. That's good. That's good. Again, for those of you who don't know, this kind of thing is really geared towards like very ambitious high schoolers and people that have background in biology, but I'm trying to make it accessible for as many people as possible, because just to soapbox really quick, and I know I do this every stream, but just for those who maybe it's your first time watching me, my goal is to teach, not to teach because that sounds condescending. My goal is to share with you biology that I find exciting, that mainstream science media oversimplifies or doesn't get deep enough into, or can't because there's too much context. And I want to be there to help with sharing the stuff that actually makes me excited about biology. So if it skips codons a bit, then it's mutagenesis. Yeah. If it skips the codon, it's not really mutagenesis, but you get different varieties of proteins. Okay, let's continue. So this is their plan, and so it's split into two, and we'll just tackle them one by one. Okay, so this is somewhat complicated, but I'll do my best to walk you through as much of this bit by bit as possible. So prion formation is deadly and allows for a readout of PSI formation. So this is basically they're set up to confirm that they are successfully making PSI protein, not only by using a fluorescent reporter so they can shine a light, and if it fluoresces green, then they'll have known that they will have made prion protein, but actually aggregating. Prions are not good for the health of the yeast. As I'm sure you can imagine, prions are not good for us. They're also not good for the yeast when they aggregate. So prions, or yeast, were hooked up to a promoter, a genetic sequence that regulates whether or not a gene is made into protein, where galactose is in the plate. So if there is galactose, this type of sugar, then the yeast will make prions. And what we can see is that when you have these 3 strains of yeast, so this first strain is a prion making yeast, this second strain is a yeast that doesn't make any prions, and this third strain down here is sup35 without the prion domain. So this yeast down here is incapable of making or of aggregating their sup35, which I think is interesting. It's a bit like how viruses have error-prone DNA. That is the analogy that I made earlier, but it is different enough to be notable. And so what we're looking at here are serial dilutions. So you basically take a solution of yeast and you dot it on this plate, and you can see the growth of the yeast. And so the leftmost contains the highest concentration of yeast, and the rightmost contains the fewest. So when you are plating on a dish that has glucose as the food source, you see that the PSI, the prion-making yeast, the no prion yeast, and the yeast that can't aggregate, they all grow about the same. Okay? I swear this matters, because it's important for us to know how this science was done. When these yeast were grown on galactose, when they start getting the instructions from the promoter to make the prion protein, you see that the prion-making yeast, they don't grow at all. They die because they're eating galactose, and that galactose is binding to the promoter, and that promoter is making the prion, which kills them. So this is just confirmation that they're successfully making and aggregating protein, which is helpful. In the yeast that started off with no prions, you also see reduced viability. So these yeast, they start off with no prions, but then when you grow them on galactose, they're not as healthy. They struggle. And then finally, we have the delta PD. So this is the yeast that is incapable of aggregating. And you see that they're completely fine on galactose, even though they're making the prion domain. They're not aggregating quite as bad. Okay, so this strategy of checking for toxicity allows us to screen for genes that are going to be encouraging prion formation and discouraging prion formation. So here we have a typical strain of yeast, nothing too fancy. And we have a yeast whose gene for the heat shock protein 104 has been removed. Okay? So why are we doing this? So remember initially, if I were to tab back, that we're trying to determine if the stress response genes affect PSI formation. And what we're doing here is we are using a library of yeast. We have 4,700 strains of yeast, each with their own unique mutation in a specific protein that disables that protein. And we're trying to check which of these genes is important for the formation of prions. And so what we see here is that the HSP heat shock protein 104, when you remove it, the yeast get a lot healthier. So what is a heat shock protein? Biologists in the chat might already know when they see HSP, because this is a very important class of proteins that makes sure that your cells aren't overrun with garbage. But the heat shock protein is something that unfolds amyloids. So this is crazy to me. This is crazy. And I'm going to pause here to explain the shock. I thought there was a misprint here. I legitimately thought that there was a typo, because this is crazy. And I'm going to explain why. Chaperones, heat shock proteins, are the thing that unfolds misfolded prions. So what the heck? So if they're unfolding bad prions and you remove them, you would expect that there would be more prion formation because HSP unfolds prions. If you remove HSP, then they're no longer unfolding prions, but they survive better. That's weird. Chat, do you get why that's weird? The thing that's supposed to be saving us from prions, if you remove it, the cells get better. That's wild, right? That's wild. I thought this was an error. Maybe the HSP also triggers apoptosis once a certain threshold of badly folded junk. Maybe, but that's not where we're going here. This is really bizarre. Why does removing an amyloid unfolder improve viability? I want chat, just like what Mandrake is doing, to hypothesize a mechanism by which this is true, because even if you can come up with nothing, and believe me, like Mandrake, you did better than me. I didn't even bother thinking about my own reason because I was so incensed that this was true. I couldn't believe my eyes. I could have sworn that they mislabeled this. But I want to spend a little bit of time to just brainstorm why this counterintuitive result could be true mechanistically behind the scenes. Because when I tell you the answer, that is going to make their genius all that more rewarding to observe, or their stupidity, whatever your angle is, right? But in order to fully appreciate their explanation, we should spend a little bit of time brainstorming this. Maybe the prion itself is suppressing apoptosis. So Mandrake, you've got a very circuit level understanding, or circuit level bias, I should say. It's okay to have a scientific bias. Thinking at the level of circuits isn't necessarily wrong. I think that's a valid hypothesis, albeit unlikely, because we know that prions are generally pretty bad. But when you have crazy results like this, you kind of have to think of crazy things. So I actually really respect that line of inquiry. The prion fragments could cause even more problems. That's an interesting hypothesis as well. So fragmentation, that's actually a different thing. The heat shock protein here, I don't believe cuts it up. So that's actually an issue with a class of anti-prion drugs that was made, where I didn't mention it in my prion video, but there is a class of anti-prion drugs that like cuts the prions, but then those prions are like double prions now. So you've just made the problem worse, and that's why that drug never made it anywhere. But that's not what we're talking about here. We're talking about purely folding and unfolding. A chaperone that folds and that unfolds aggregated prions. I'm going to take a sip of my water, and then we will continue and see what the authors hypothesize is the issue here. I would call it an approach. Yeah, not a bias. Sorry. It's an approach. It's an approach. Very appropriate from systems medicine to think at the circuit level. Does HSP also act on non-prion proteins, or is it just prions and amyloids? I believe HSPs are generally pretty multifunctional. But I would not be able to tell you about HSP 104 in particular. I'm not really a chaperone expert. That's a good question, though. Okay, so let's see what the authors come up with. And I like their explanation because it is kind of crazy. but I like that. It's interesting, and not only is it crazy, but to me it's kind of believable. So this is unfortunately a paper that is paywalled. Boo, paywalls. But this is the abstract, which I can show. So let's just read this together. This is their abstract for a different paper where they're trying to explain this phenomenon. The yeast non-Mendelian factor PSI has been suggested to be a self-modified protein analogous to mammalian prions. Here it is reported that an intermediate amount of the chaperone protein HSP104 was required for the propagation of the PSI prion factor. Overproduction or inactivation of HSP104 caused the loss of the prions. These results suggest that chaperone proteins play a role in prion-like phenomena, and that a certain level of chaperone expression can cure cells of prions without affecting viability. This may lead to anti-prion treatments that involve the alteration of chaperone amounts or activity. So removing the chaperone and making too much of it had the same effect, which is absolutely crazy. Yeah, it's crazy. So why? How? What is the mechanism that they are suggesting? So in that paper, and I did read the paper because I have access, unfortunately you don't, they argue that the prion bound to chaperone may form a prion template that induces aggregation. What? Chaperoning it right into an aggregate. Incredible. Yeah. So here's one way that this can turn out. Right? Well, here's one pathway. So you have your misfolded prion, very sad, binding to the chaperone, and then it unfolds, and then it releases that misfolded prion, and it's a happy prion again. But here's the thing. In between misfolded and correctly folded, there is an intermediate fold. And so the authors suggest that what's actually going on is that there's another pathway by which a misfolded prion can bind the halfway folded prion and cause misfolding. So, not only does the prion unfold the protein, but it also holds it in an intermediate state that then can be misfolded. So this, I think, is a very cool explanation, where if you just pump the cell full of the chaperone protein, it'll grab onto every misfolded protein it can and then unfold it. And so you can really increase that value and misfold everything, push the equilibrium all the way to this state over here. So the prion has a partner HSP, sort of. Well, I don't actually know, because I'm not an expert in this field, whether or not this is accepted, whether or not this is a popular view, but it is cool. And I like that. I think it's neat. I like reading papers. I don't claim to be an expert in any of these fields. I'm just a fan, I guess I should say. So if you remove the equilibrium here, if you remove the chaperone, if you remove the chaperone entirely, then you no longer have the capacity, the halfway folded prion, that the misfolded prion needs to bind to misfold it. Interesting, but how would you test that explanation? That's such a good question, Simon, because I don't know if Clockwork is still here, or any of the structural biologists here who can offer some extra brain cells. I really don't know, because these intermediates are very short-lived. It'd be quite difficult to test. At least I can't come up with anything, but it is an interesting question. Okay. So hopefully that kind of gets across how crazy, like, we're only halfway through figure 1, and I've already had at least like 5 brain blast moments, like figuring out what this paper is about. So this HSP, we need an intermediate amount for the HSP to hit the sweet spot of having a normal level of prion, but then when you remove it, as the delta HSP does here, you get no prion formation, and then that means you get more yeast growing. So that is what we're looking for here. So all the genes that we're screening in that library of 4,700 yeast, we're going to split them into suppressors of toxicity. So HSP, suppressor of toxicity. When you remove it, they get happier. The other class is enhancers of toxicity. So this is a protein, HAC1, which activates the unfolded protein response, or UPR, which if you have seen my prion video, you would know that that is the program by which the, oh God, what's the name? Endoplasmic reticulum starts pumping up the machinery that is needed to unfold misfolded proteins. So this makes sense. Unlike the HSP, which completely blew my mind, the protein that activates the unfolded protein response, if you remove that, if you disable that protein, then you get more prion production, which means fewer healthy yeast, and that makes sense. All right. Hey, keep doing such videos. They are very helpful. Wish more YouTubers do some streams/videos. If you want to check out VTheorem, links in the description. I have some other VTubers who also do content in their fields. yeah, I think biology right now on YouTube is experiencing a renaissance, and I've talked with Clockwork quite a bit about that. So hopefully we'll get a bigger piece of the YouTube market share. We can only hope. Okay. So we have our tool. We are going to test 4,000 plus yeast, each containing an individual mutation in a particular gene, and we're going to test which ones help prion formation and which ones don't help or reduce prion formation. And we're going to do that by seeing which yeast are better at surviving and which yeast are worst at surviving, comparing between the prion form and the non-prion form. So walking through their figure here, we have an initial screen with this deletion library. So that's this YGDS, that's the deletion library. And then what they found was that there were almost 400 deletions that enhanced toxicity and then 350 hits that reduced toxicity. And you can see samples of the yeast here. And then when they did a bunch of quality control things that I'm not going to get into because I've talked a lot already. And this is more like, I think, Resta's field of interest, where we're thinking about how to computationally control for good data analysis, which I won't get into because I'm not going to be able to do justice there. I'm just going to assume that they did this part correctly. So what did they find? So here we have Go terms. This is something that Resta probably very familiar with. Basically, we are looking at all of the genes and we are grouping them by their function. And then we are going to see which ones are more active we are going to see which ones were better at reducing prion toxicity and which ones were worse at reducing prion toxicity. So we have a lot of different categories here, and we are comparing the library, which is the mutation, the yeast mutants, to the yeast mutants that have had the prion treatment, as we'll call it. So the red is plus prion, and the blue here is minus prion. QA is my field. Sorry. You can go check out the stream I did with Resta if you want to talk about control of data, because it's just not my thing. But I respect people who do it, but I wouldn't be able to explain it very well. So chat, is there anything on this list that maybe stands out to you? Well, just pause on this screen while I take a sip of water. Have the experience of looking at data without my help to interpret? Is there anything that is interesting to you? Actually, it doesn't show up very well on the slides, but just looking through this, anything surprising? I'm actually looking at this list again, and I've just found something else that I found to be quite interesting, which is really, really, really weird. Bioprocesses unknown, organelle COP, transportation. Yeah, so if we look down here, towards the bottom, we see that the library, so without the formation of prions, something's fragging out here. There's a surprising number of genes whose functions we don't know in yeast. And I'm not like a yeast guy, so maybe this is only surprising to me, but I kind of figured because we like basically eviscerated E. coli and know E. coli from top to bottom, and that's not really fair because we're still discovering things about E. coli every day, but like we know so much about our favorite model organisms that it's just kind of surprising that there are so many genes that are in biological processes unknown left. Organelle comp, yeah. Organelle organization and biogenesis. So there are a lot of the yeast that reduced prion toxicity. There are a lot of them here that have had these genes upregulated. So that's pretty interesting as well. I want to point out to two different categories. So here, response to stress between the yeast that reduce toxicity and just the mutant ones doesn't seem to be higher than the library, which is kind of interesting because our whole thing here is that we're activating the stress response, and stress response doesn't seem to be crazy different between these two populations. The other thing that is kind of weird here is protein folding. You would think that prion things would activate protein folding, but doesn't really seem to be the case, which is kind of weird. Do you have any idea about protein expression via yeast? Is it better than bacteria expression systems? So that's kind of not related to the stream, but to keep it brief, they're good at different things. You can do things in yeast that are fancier than in bacteria, but you can do things in bacteria that are maybe easier to do than in yeast. Okay, so now we're looking at the other side of the coin. Genes that enhanced toxicity. So before were the genes that reduced prion formation. These are the genes that improved prion formation. So yeah, enhanced toxicity, improved prion formation. This is the interesting one, I think. Anything that we notice here? We still have that biological processes unknown being really high in the library, but now the red bars to me look a lot more interesting and a lot more worthwhile. What's crazy here? That the stress pathway isn't number one. That's not necessarily a bad thing because there might just be fewer stress genes. So we have to keep in mind that, you know, actually, I'm not entirely sure. I should have looked a little more carefully as to what the x-axis here is, this percent number. If someone has the paper pulled up, they might be able to inform me. But I wouldn't necessarily, it's not about the size that matters here in this case, I think. Now, what I find to be interesting is comparing this graph with the previous graph. In this graph, the ones that enhance toxicity, the ones that promoted prion formation, are actually upregulated in response to stress, as opposed to the reduced toxicity graph, which showed basically no difference between reduced toxicity and the library. So to me, that's really interesting. And indeed, that's what the authors want us to focus on. The fact that the things that make prions form, that is controlled by the stress genes. I just find it weird that you don't see the other side of the coin, right? In response to stress genes are turned on. Those stress genes help these prions form. But in times of stress, these stress genes don't actually prevent prions from forming as well. I would assume that you would maybe not want the prions to form in certain cases, but maybe this is a unidirectional thing. Maybe it's kind of stupid to expect it to go both ways now that I say these thoughts out loud. Some of the same gene types are those organelle ones. Yeah, and I really don't have an explanation for that kind of thing. Signal transduction is a large jump compared to the 350 or so. Yeah, to me as like a molecule person, when I see high levels of change at the level of signal transduction, to me that can mean like anything. Signal transduction is just everything. And maybe that's offensive to the signal transduction people, but it kind of is everything, which is why it's so important. But when I see that, I don't know, I'm not surprised, I should say. Okay. So this is great for the author's hypothesis, right? The genes that are responding to stress are upregulated in the yeast that are better at making the prion. So we can then take a look at a list of promising candidates. And so they've narrowed down that initial list of like 300 genes down to about 40, and then even further here. And we can test individually, now that we've singled these yeast out, what their ability to make prions is. How good are they at making prions, basically. And then we can categorize them by what they actually do. So on the y-axis here, we have each individual gene that they've each individual gene that they have singled out. And on the x-axis, we have the fold prion induction frequency. In other words, compared to a yeast that doesn't have any of these mutations, how much more or less prions do they make? Okay, so I don't know any of these genes off the top of my head. We see HAC1 here, and that we saw earlier in their figure. And then we see PPQ1 here. This is crazy to me, and I don't know if the authors addressed this, but the gene with the most prion inducing power the gene that most influenced the production of prions, has other as a function. I don't know what PPQ1 does. If anybody wants to look that up, that would be very interesting. But it's surprising that the number one thing that makes prions happen in this yeast isn't even like a folding protein. It's kind of bizarre. And then down we have here, so that's like 10 times more prions that are being made if you delete PPQ1, whose function I don't really know. But everything else kind of makes sense, right? MSN2, so this one is yellow, so this one's in the stress response pathway. That's awesome, we like that. NOT3, so this is orange, so this is the UPS, this is ubiquitin proteasome. So the proteasome is sort of the cell's garbage disposal. It chops up proteins. So it makes sense that if you're trying to get rid of bad protein like the prion, that you would upregulate the UPS pathway. Here, HAC1, we have the UPR gene, so the unfolded protein response. So this makes sense, and it's really helpful for their hypothesis that some of the best ones at making prions are genes that are involved in these stress response and folding pathways. It's a signaling kinase, PPQ, a novel protein phosphatase containing serine and asparagine, rich amino terminal domains involved in the regulation of protein synthesis. This is why signaling to me is very hard to get into, because that doesn't really say too much to me right away in terms of connecting the dots here. Interesting to know, though. It's one of those signaling proteins. Okay. So I would say, and you can disagree in the chat, that the authors did a pretty good job at addressing this first experimental goal. So they wanted to determine if a stress response affects prion formation, and they found a whole bunch of stress response genes that affect prion formation, and they're pretty good at making prion. Makes sense to me. The second part is the part that I'm more interested in, determining whether or not the actual stress affects prion formation. So here's how they do it. They grow wild-type yeast that have some baseline of prion just hanging around, and yeast that don't have any prions, so clean yeast in stressful conditions. And then they grow them both on food-rich plates and plates that kill yeast without prions. So this is called a selective plate. So the reason why they do it on two separate plates is that this first set, the non-selective media, so this is just a normal buffet for the yeast. They have all the nutrients they need. They're very happy to grow on this. And by doing serial dilutions and counting the number of colonies, you can get a sense for how well the yeast survived that challenge. I won't get too deep into that technique. Just if you're not a biologist, just trust that this left-hand side, if you count the number that is on these plates, you get a measure for how well those yeast survived the challenge. And on the right, we have a plate that is specifically formulated to kill the yeast that don't have prions. So this allows us to measure basically prion formation in those specific yeast. So we have the MSN here that we saw in that previous slide. So if we compare it to wild type, wild type, they're okay at making prions, they're baseline, vanilla, you should say. And so they have a certain number of colonies. But MSN2, which we see here, MSN2 makes 10-fold the number of prions. And if we count the colonies here, this is likely about 10 times the number of colonies in the wild type. So that's how we're measuring both how well did the yeast survive challenge, as well as how much prion did the yeast make. Just a wild idea to find intermediate prion binding configurations. Again, I'm not a structural biologist, but folding intermediates are often really challenging to find because they are by definition not stable. So it would be a legitimate line of inquiry to see whether or not there were scientists that attempted to stabilize an intermediate folding of a prion, but I wouldn't be surprised if they all failed too. Okay, so hopefully we kind of understand that this is a vague readout of our two metrics, right? Fitness, which is the number of colonies that grow on the left, and the amount of prion, which is the colony on the right. So it's not the absolute number of prion for biologists who are going to catch me by not explaining it completely correctly. We're counting the number of yeast that can make prion. So we're not measuring protein level, but the number of yeast that make prions. Okay. And so we have this monster of a graph, and I'm sorry in advance, but I will do my best to walk you through bit by bit to explain their data. This is the data. It's kind of overwhelming, but I'm going to put a white box over the majority of it so that we focus on the graph one bit at a time. But just to see the overall structure of this data, on the y-axis here, we have the cell number of control. So this is the viability. So this is how well did the yeast survive the potential challenge. And on the right, we have fold PSI induction frequency. So this is a logarithmic graph, 1 down here, 10 times up here, and then 100 times up here. So this has two y-axes. The orange one is this left axis, and the blue bars correspond to this right axis. On the x-axis, we have all of the different conditions that these yeast were put through. This is sort of the ringer, the deadly challenge, we'll say. And so now I'll go through the data bar by bar. Flash freeze with liquid nitrogen, freeze dry, find gold particles with SEM, go to atomic force microscopy, scan the prion configurations, yucky and complicated. Yeah, that doesn't sound very fun, and prions are not... Again, I'm talking out of my depth as someone who's not a structural biologist, so you might be able to help out, but since prions aggregate and they don't really form regular patterns, I wonder how useful that approach would even be, because also if we're talking about major mammalian prion protein, they can have different lengths as well. I mean, I guess you could build up a database of different intermediates for different fragment sizes and different configurations. That would still be better than nothing. It would be fun though. Yeah, it would be cool. And I think the reason why I never got into prion science professionally is because structure stuff is so complicated. Although it might pain clockwork to hear that, I do find it to be quite hard. But I love the science that it produces. It's so cool. All right, let's look at the data. So first things first. This data is organized in descending order. So from left to right, we have the biggest survivors in orange, and then on the right we have the most prions produced. So that's why they're ordered this way. So that's why the x-axis is going to be kind of confusing. So let's start with the control. The control is no challenge, just grow normally. So this is normalized to 100%, which is why the orange bar is up here. Again, yeast that are just hanging out have some baseline level of prions, which is why there's some prion amount here, and we normalize that to 1 down here. The first thing that the authors want to clarify here is that we are measuring stress and not death, because you can imagine that death and stress are very correlated, right? If you stress a cell out, it dies. If a cell dies, it was lightly stressed. So there's this correlative factor here that we have to address. And so the authors grow their yeast in two conditions that are, according to them, conditions that do death but don't do stress. And that would be these boxed conditions, no sugar and H2O. I think I missed a slide title here. So this title should be that when you starve yeast by not giving them sugar, or when you grow them in a not friendly, not salty environment like pure water, they'll die. They won't grow, but they'll enter what is known as a quiescent state, or a sleeping stage. If you've watched my tardigrade video, it's like the ton state, right? They change into a form where they are much more resistant to stress, but it's not related to the stress genes, weirdly enough. So that's what these are here for. And we can see that even though we have death or lack of growth in the no sugar and the water controls, we also don't see more prions. So it's not that like death causes prions. It's more like if we find an effect, it's most likely due to stress. So the first stressful condition is 1 molar potassium chloride. So this is quite a high level of salt. 1 molar is quite high. But yeast, I don't know a lot about yeast. 1 molar salt is quite high for the stuff that I work on, which are very fragile mammalian cells. I don't really even know if E. coli can tolerate 1 molar salt. But apparently the yeast love it. I mean, you have more than 100% growth in the 1 molar potassium chloride condition. But this condition doesn't produce any prions, so it's underneath the control. Then we go to the next, 30 degrees Celsius. I don't actually know why this is basically the same data as the control. I think it's to remind the viewer that the control is the yeast are grown at 30 degrees Celsius. So let's just call this control part 2. 30 degrees Celsius is their happy zone. So 4 millimolar DTT, so this is a molecule that destroys proteins. And it hurts. Full 4 millimolar might not be that much DTT, but you do see a bit of a reduction, maybe 10%. That doesn't increase prions. 1 molar sodium chloride, so this table salt, that hurts more than the potassium chloride, interestingly enough. And still no significant difference in terms of prion production. Hydrogen peroxide, 5 millimolar. So this is a DNA damager. It's an oxidizer. This is a nasty chemical that destroys biological molecules. And you do see a reduction, 25% of the control. Still no prion production. Another salt that hurts a lot and increases, it looks like prion production, but this difference actually is not significant. 39 degrees Celsius, so just a 9-degree change in Celsius temperature. You see a ton of cell death, but still no prion production. So it doesn't look too hot right now. It doesn't seem like this is a very common method to adapt to stress, but we'll continue. 1.5, getting saltier. So this is very salty. And now we're starting to see something. So we have about 30-40% survival. And we have significant increase in prion production. So if you subject these yeast to an insanely high amount of salt, then they start making prions, right? So at control, you're down here at 1, and you dunk them into very salty water. Maybe this is, I don't really know how this goes, maybe threefold, but it is statistically significant. Okay. We're getting worse now. So lithium chloride, very salty, increased prion formation just like sodium chloride. 42 degrees Celsius, very hot, and production of prion. So the worst conditions that we're getting at, just like we're just slapping these yeast, boiling, not really boiling them, but 42 degrees Celsius is very hot for a yeast. And we're seeing that as these conditions get more severe, that you get prion formation, although not always. It's important to note that in this particular condition with NH4 acetate, you don't get prion formation, although I wonder if that's because this particular molecule itself inhibits prion formation. It would be interesting to know. I'm going to ignore some of these. They're testing more heat conditions. 43 degrees Celsius. Now we're getting so low. And maybe this prion difference isn't significant because you just don't have like enough surviving cells, which is possible. 12 millimolar DTT. So DTT, as I mentioned earlier, was the molecule that destroys proteins. And when you pump that up to 12 millimolar, very high concentration of this protein destroyer, you see that you get a significant loss in cell viability and an increase in prion formation. And these two dots here, these indicate that the yeast that make prions survived better than the yeast that don't make prions at all. So when you add 12 millimolar DTT to both prion-containing and prion non-containing yeast, the prion-containing yeast actually do end up surviving better, which is why there are these two dots here. So that kind of is one example of the author's hypothesis being true. And we'll continue on with the rest because I can't be bothered to do them. But you start seeing, as you get much more severe in terms of what you add, you get a lot of prion formation and not a lot of viability. But in total, if you don't see a dot on this axis, it means that the prion containing and the prion non-containing survived at about the same rate. If you see one dot, it means that the prion non-containing survived better than the prion containing, and 2 dots means that the yeast that produced prions survived better than the prions that didn't produce prions. And I know it's a very long mouthful, but I want to point out here that in the majority of cases, it didn't seem to matter, right? There are not a lot of dots down here. It didn't seem to matter in the majority of cases whether or not a yeast made prions. If you subject them to an insanely high temperature like 43 degrees Celsius, it didn't matter whether or not the yeast made prions or if they didn't. They all died about equally the same. In some conditions, it actually helped that the yeast didn't make prions, like in the lithium chloride condition and manganese chloride. But in 2 of these conditions, 12 millimolar DTT, and there's one more somewhere, 2 molar potassium chloride. In these 2 conditions, it did appear that the yeast that grew with prions survived better. And that's kind of nutty. In general, though, as my hesitance or skepticism at the beginning of this presentation warranted, generally making more prion is correlated to worse survival. It's correlated, it doesn't necessarily mean that it causes worse survival, but we know what prions do in us, so it's not that big of a leap to make. Okay, I think it's pretty safe to assume that the authors also hit this point out of the park. That, yeah, I buy with this fold change that something as stressful as 2 molar lithium chloride made prion. And so their argument for this evolvability thing is that prions react in response to stress genes, and they also just respond to stress, right? And that is evidence that the prion protein may have evolved to hold on to genetic variation and unleash it in times of stress so that the yeast can adapt. And I want to take a second here to ask the chat, first, how are you doing? That was a very complicated graph, and if you tuned in a bit later, you might be a little lost in the sauce, which, is okay. How are we feeling about this paper? And what do you think about a level of evidence that the conclusions here have for this initial model? Right? The model is this thing up top here. Yeast are exposed to environmental stress. You have this termination factor called the sub-35 that gets pulled out by aggregating with prions, and then you start ignoring stop codons, and by ignoring stop codons, you get a lot more expression of different proteins, and that causes better survival in extremely stressful conditions. That's the model. And we have these 2 experiments, and I'd say, from my perspective, that the authors did do a good job at proving these particular points. But in terms of moving the needle on your opinion as to whether or not this is true, do you think, like how would you describe the contribution of this paper to the field? And you are free to ask me any clarifying questions by what I mean by that. Because at the end of the day, when you are a scientist and you finish reading a paper, you have to kind of determine how important is this paper. From what I got from the graph, I don't see how PSI helps this yeast. It's really unfortunate because there are only so many dimensions to showing the data, but basically you kind of have to take their word that these 2 dots mark the populations. It's not obvious from the data here, but these 2 mark the populations that had improved survival when you compare prion plus and prion minus. Whereas this graph is yes stress or no stress. So it's actually not really shown on the graph. It might be in the supplemental, but I actually didn't check. So Alexandros says, good start. Any other opinions? Just looking at this here. And so what I want to help my audience develop is a sense of putting science literature into its context. Because, and I know I rag on Reddit a lot, but there are a lot of times, especially in the field of psychology, where there'll be a paper posted to R Science and there'll just be comments just completely trashing the paper. And in some cases, that's warranted. But in others, what the feeling is just maybe the paper isn't as strong as you would have liked. Or, you know, and that's okay. Not every paper is going to be a Nature or a Cell paper or a Science paper. Not everybody gets a home run every single time. But that doesn't mean that these papers aren't important, or that they aren't going to be used for important things. So I don't want to train an audience to just go, oh, correlation is not causation, and blindly criticize science that way, but to form sort of a skepticism, an internal skepticism and a higher order feeling for science as a field. And part of that is coming to a conclusion about how we feel about the paper. Well, the model is at the very least a lovely idea. I definitely want to see other labs confirm the model via other approaches. That's true, that's true. How would you confirm the model via other approaches? That's kind of what I'm getting at. Because what I'm leading at is not a criticism of the paper. Because I think the paper is quite nice. It's been cited a whole bunch of times. And this lab is, from what I gathered from reading the papers and the number of citations, a pretty high-powered, important lab. So I'm not going to say that it's criticism, but it's important to acknowledge, to keep the paper in its context. So systems, what other approach? Why do you need another approach? Is a good discussion question. And I'll maybe get into a little bit of a soapbox here as we're thinking about how we feel about this paper. It's okay not to have an opinion or to be lost. That's also okay too. It's complicated. It was complicated for me. So if you were able to somewhat follow along, I would consider that a win. But here's something that I want printed on a shirt, maybe for merch one day. It's that models are meant to be broken. And this paper, my advisor used to say this all the time, and I credit my advisor for helping shape the way I think, training me to think like a scientist. And I think what's important to acknowledge is that this paper doesn't really try to break this model. And what do I mean by that? It's the bar, I think, is kind of low for aim 1 to be a success. Especially because, as some of you pointed out, stress response wasn't like the top of the list, although it happened to be that those were the ones that were very good at forming the prion. But when you have a model like this, environmental stress to prion aggregation to nonsense suppression to survival, you ideally want to design an experiment that tries to break this. And when you try to break a model and it stands the test of your scientific brain, that to me is better. That doesn't mean that this paper isn't important, but as you'll see here, determine if the stress response genes affect the PSI formation. That's not really trying to break anything. Because when you screen genes, you know, there's some chance that you just get a stress response gene that shows up in the screen anyway, just because screens can have all sorts of junk in there, and that kind of junk might not be very important, right? And what's also important to know, and again, this is taking this paper in a vacuum outside of its context, which is something that you shouldn't do, but in terms of if I were to just read this one paper, I know nothing about whether or not those yeast survived better because of nonsense suppression. Right? What have we actually learned about this model? We learned that there were two things that we assumed were true in this model. And we learned that those assumptions are true in a very broad sense. But you could equally interpret the data as saying, oh, more PSI is sometimes good for the cell, because we didn't test if it was nonsense suppression. Does that make sense, chat? we didn't try to see if nonsense suppression was the thing that was important. We checked two things. Or rather, I think, let me walk that back. This second one, I think, does try to break this idea. They're trying to see, testing a whole bunch of different stress conditions, whether or not stress can cause PSI to trigger, Prion to trigger. This first one I think I have, the first one to me is a little more wishy-washy. It's nice. It's nice to understand that stress genes affect the prion protein. And experts in the field may disagree with me because I'm not an expert. This is my naive, educated opinion. It is how I form my understanding and evaluation of this paper. I'm not saying that I'm right or that you should take my word as gospel, but I'm modeling that that is how I feel when I evaluate a piece of information like this. It's jumping to its own conclusion, basically. I wouldn't go that far. I would say that it gave itself a layup. It doesn't feel like the authors are rigorously trying to break their own model. It just feels like they're trying to find things that are consistent with it, as opposed to trying to break it and then finding out later. For example, something that I thought of just now is, what if we did an experiment where we added something to stop nonsense suppression, where we have the prion aggregation and we do all this battery of tests, but then we try to stop nonsense suppression. I don't know how, it might not even be possible, but if you try to stop nonsense suppression, do you still get that survival? And that is what I mean by breaking that model. So if this nonsense suppression is true, then you get survival. What if you don't have nonsense suppression? What if you try to break this? I hope that makes sense. All right, let's move on to a summary. So this protein, sub35, is a chaperone protein that helps the cell stop elongating a protein chain when you hit a stop codon. The prion form, PSI+, is a nonsense suppressor. It helps the cell ignore those stop codons. PSI can confer a selective advantage, but it's rare. Ignoring stop codons is generally a bad idea, and this is a citation from their previous paper that had shown that this PSI prion can be selectively advantageous 25% of the time. If these prions are made in response to stress, it could be a mechanism to quickly improve evolvability when times are bad by bringing out genetic diversity that was hiding behind the stop codon. And this paper shows two features of an evolvability mechanism that the prion displays. One being that the genes that control the stress response also control the PSI formation, as well as the fact that stress as an inducible thing you could do also causes prion formation. Yeah. And sometimes, in those two cases with 12 millimolar DTT and 2 molar KCl, sometimes having that prion is actually a good thing, that those particular yeast made something potentially in their scramble if we think that the model is true. But again, because they didn't break the model, we don't know if prions are actually just good at counteracting potassium chloride toxicity or counteracting DTT toxicity. They didn't try to break that, and I think that is something to keep in mind. It's that level of skepticism that my advisor used to say, like, don't drink the author's Kool-Aid, come to your own conclusions. And when we say come to your own conclusions, that's what we're saying, is you want to be nice to the authors, you want the authors to write a good paper, but then you take your own critical thinking and identify where those gaps are. And there will be gaps. There's no perfect paper. I don't want to seem like I'm like dissing the paper. I'm absolutely not. This is a great paper, but you don't want to take away too much. You want to take away just the right amount for the conclusions for the paper. I'd like to see the work pseudo-reproduced in an E. coli lab. I don't know if that would work because we have different ribosomes between eukaryotes and bacteria. Although that's an interesting idea. Also, I think bacteria, I think some of them can use different stop codons. It's a total mess and I don't quite remember at the top of my head. Okay. So, what do you think? I think I asked this question already, so I'm just... Well, So the thing. So the reason why I have this slide is to take a look at what my perfect experiment would have been. And now I know, having talked about this out loud, that this thing about mass spec might not be like a winner, but as a commenter had pointed out, maybe an RNA sequencing thing might have worked. But we'll see that like checking if the weirdo proteins are sufficient to improve fitness by putting them into prionless yeast, that's the thing that I'm thinking of that would break the model, right? If the model is these yeast make weirdo proteins that have extra bits at them because they ignore their stop codons, why don't we take those weirdo proteins and put them into yeast and see if they can survive that stress? That would have broken the model. It would have been an attempt to break the model. And since this paper is in 2008, I don't actually know if there's been follow-up, but this is the paper that I could get that I could share. Again, I'm limited by what I'm allowed to show you. It'd be cool to do a follow-up one day, but yeah, that's where I ended my research for this particular experiment, or this particular paper. So we are at the end of the paper. I'm happy to take any questions, as well as just chat very briefly with you all. It's been a while. I haven't been streaming because I've been working on some stuff behind the scenes, which have been pretty exciting, as well as a series of YouTube shorts that I hope will help to describe some of the techniques in the MolBio lab in a way that is fun and engaging. So if you haven't seen my most recent short, why don't you check it out? It's about Western blots and how we measure protein. Yeah. I know that classes have started for a couple of you, and have been in session for a while, depending on where you are in the world and what exactly your school situation is. For me, it's not too far off as well. I won't say exactly when my start date is, but I am savoring the last bits of summer before I go back to teach students. Because like doing this hobby, I think keeps my skills as a biologist sharp, which I think is interesting. Because without this, like, I would be mostly reading stuff that I am required to read, but by reading for this channel, I actually get a lot more exposure to a lot more fields of biology, which I think ultimately helps me professionally. I actually recommended, because I had learned about the toxin antitoxin thing from a previous video, to make a video on that, and I realized that that could be a potent engineering tool. And so that was now a path I could recommend for students who are looking to get into genetic engineering, which has been pretty interesting. I'm glad I blundered into your channel. You made a tough day a happy day. Bye. Thank you for tuning in. I hope you enjoyed my presentation on prions. But yeah, I think that like I'm much more suited to read broadly than I am to read specifically. Like reading in my particular field and as a graduate student was really engaging, and I loved getting that deep in there. But I think as a graduate student, you get so wrapped up in your own thing that you miss out on a lot of other cool biology. So I like being in the position to just kind of sample the whole landscape. It's been really, really rewarding. I know that there are requests to do other prion-esque content like Alzheimer's and Huntington's disease, and I think I'm really not ready to talk about those at any time coming up, just because those are really, there's a lot of research out on Alzheimer's. Of course, there's been a huge push in funding to do Alzheimer's research, and unlike prions, where the field is relatively smaller than Alzheimer's, if I get something wrong in the Alzheimer's field, I'll be like instantly deleted from the internet. It's also touches upon like the field of diseases that I'm kind of unwilling to cover because they do have such a high cost of human suffering across so many different people. And I'm worried that covering them insensitively would result in unhappiness for the channel, I should say. But I think one day I'll be ready to do Alzheimer's and cancer and all those other things. Just not now. For those of you who don't know and who have stayed till the end, I have a Discord link in the description if you wanted to join that. I always wait sort of until the end or in the middle of the stream to try to catch the people who have stuck around for the majority of it, to sort of stem the tide, because I'm a little afraid of opening the Discord to everybody all at the same time. So if you have more interesting thoughts about prions that you want to let me know, or let me know how the stream went, feel free to leave a comment in the Discord or in the comments in the YouTube section. That's also very good too, and it helps the algorithm supposedly. All right. Well, I've done quite a bit of yapping. And I think for me, it is now time to get going. I hope you all have a lovely day and learned a thing or two or three about the weird world of yeast prions. And I will be uploading another short tomorrow about scanning, alanine scanning mutagenesis. So keep an eye out on your shorts feed for that. Bye, y'all. Bye.